Thank you, Andy. It's a pleasure to be here. I'd like to thank the organizers of the meeting and the board from the LRF and the executive leadership from the LRF for inviting me to speak to you this morning. It's a great pleasure for me as a pathologist to be able to talk to patients about what we do. It's also a great pleasure to be associated with the Scientific Advisory Board, which is really doing great work to advance both the science and the treatment of lymphoma. So I think I have a question coming up, if I can advance the slide. There we go. Okay, so you have to read the fine print, but this question is really asking, what kind of lymphoma do you have? So if you can read that, you can read them out loud. You can type in the answer. A is for CLL, small lymphocytic lymphoma. B is cutaneous lymphoma. C, diffuse large B-cell lymphoma. D, follicular lymphoma. E, Hodgkin lymphoma. F, mantle cell lymphoma. G, marginal zone lymphoma. H, peripheral T-cell lymphoma. I for Waldenstrom macroglobulinemia. J for the group of T-cell lymphomas. K if there's some other B-cell lymphoma we didn't mention because, as they said, there's like 80 kinds. And L, I'm not sure yet because pathologists are taking too long, maybe. All right, so we're biased towards the indolent lymphomas with a lot of follicular lymphomas. Some, as we mentioned, large cell lymphoma is very common, so we have 10% large cell lymphoma, and CLL, chronic lymphocytic leukemia is common, so we have about 10% of those. Mantle cells coming up there at 14 or 15%. So a nice distribution. Okay, so what are my goals here? I'm going to give you a behind-the-scenes look at lymphoma diagnosis. Give you a little bit idea of the principles that we use to do this and the tools that we use to do this. And so that picture, if you're familiar with the Willy Wonka book and movie, pathologists, I think, view themselves as the Oompa Loompas of medicine. We're sitting there toiling in the basement of the hospital and the laboratories, answering the phone calls of why it's taking so long to make your diagnosis. But I'll show you why. So as Dr. Evans mentioned, modern lymphoma diagnosis and classification has really evolved over the last couple of decades. And it was really essentially a purely morphology-driven classification, even at the beginning of when I was training. And now lymphomas are classified based on not only morphology, but the immunophenotype, which is the kind of molecules that we see on the surface or inside of cells to help us classify them, and certainly the molecular genetic changes that we see that Dr. Evans alluded to. This has really been built on the back of our increase of our knowledge of the basic immunology and the molecular genetics of lymphoma. So I use this pyramid to kind of explain how, what things are based on. I mean, the histopathology or the morphology is the basis for everything. And then on top of that, we build the immunophenotype on top of that. We build the genotype, or what kind of genes that are important in lymphoma diagnosis, on top of that, to arrive at the very top of that pyramid, which is the ultimate diagnosis and classification. So what happens to the tissue when you have a biopsy? On day one, you're having your procedure. The biopsy is done. At our place, we, when there's enough tissue, do frozen sections, which means we take a little piece of that tissue, we snap freeze it, we make a tissue section of it, and get a quick read about, number one, do we have enough tissue, we think, to make a diagnosis, because nowadays you need some tissue to do some of these studies, and can we triage it then to the right studies. Then we begin the routine processing, which means fixing that tissue in formaldehyde, usually, and then if we think we need flow cytometry, we can also send a piece off to the flow lab to start that whole process. The next day, the routine histopathology is coming out. Overnight, the tissue has been fixed and processed, and slides are made. And a lot of times the flow cytometry is back, actually, at the time that we're reviewing the H&E, which is the basic morphology slide, and we can sometimes render a diagnosis right then and there. Many times we need to do additional studies. This paraffin section, the immunohistochemistry, which I'll show you, which is part of the immunophenotyping, that takes essentially another day. In complicated cases where we need to do molecular genetic studies, that may be around 10% of the cases. That can take some additional time depending on how busy your lab is and when you run those tests, and that can take anywhere from just a couple more days to, say, a week for that thing to come back. So this is kind of just show you there are lots of people involved in this process. You can see on the left people at the microscope. That's us looking at frozen section to understand, do we have enough tissue, and is this biopsy actually a candidate for being lymphoma? And if so, then we can start sending off the appropriate tissues. One goes to the flow lab, the other goes to the histology lab, and then there's people that come in very early in the morning. Our histology lab opens at 4 in the morning, and for the processors are done, and the technologists start preparing the slides. They embed the tissue in wax, then they cut the slides, they stain them, and they prepare it for the pathologist to review. So at that time, we're putting together both the flow cytometry data and the histology data to start the diagnostic process, and we can sometimes be done right there. But some of these other things, as I mentioned, do take time. Immunohistochemistry, there's a picture of our lab up in the upper left, and more molecular studies like cytogenetics and FISH down on the lower left. We have to do other molecular studies on the right. That can all take time. So as Dr. Evans noted, the framework for the classification really is a combination of history and sort of biology. So as we mentioned, 1832, a long time ago, Hodgkin lymphoma first being described. We now know that's actually a B-cell lymphoma, but it's a very special kind, and we keep it separate as Hodgkin lymphoma. The whole big, huge group of non-Hodgkin lymphomas, we know biology that there are B cells and T cells, and so we split lymphomas into B-cell lymphomas and T-cell lymphomas. And now we know, and this is the laundry list of the 80 different kinds of lymphoma that are now in the most recent classification, and so these are the kinds of things that we are dealing with now to make it a little more complicated. As Dr. Evans noted, we basically try and take what we know about normal immunology and place those lymphomas into what look like normal cells. And so the point of this slide is just to show you the different kind of things that we do. That big circle in the center is the germinal center, where B cells are developing and learning how to recognize the antibody and the antigens that they're going to recognize to make antibodies. And we know there are certain markers that those cells express and certain ways that these look and where they occur in the lymph node. We use all of that information to say, oh, does this lymphoma look like what we know to be a germinal center B cell? And if so, then it might be a follicular lymphoma. And the whole process is similar for all of these different kinds of B cell neoplasms that you see listed there in yellow. But some lymphomas, we think we know a lot, but when you really look at the names, it shows you we don't really know a lot, because some of these things are named specifically just by the way they look. So, for example, the T-cell lymphomas, there's still a lot of progress to be made in understanding the biology of T-cell lymphomas. So if you look at just the names, angioimmunoblastic T-cell lymphoma, sounds like we know what we're talking about. What does that really mean? It means like, oh, that's a T-cell lymphoma that has lots of vessels and big cells. Okay, so that doesn't actually, when you break it down, it's not that sophisticated. Peripheral T-cell lymphoma, not otherwise specified. That's really a wastebasket of things that, well, it's not specific kinds of T-cell lymphomas that I know, so it must be a T-cell lymphoma of some other kind, and we just throw it into that bucket. But we've learned things over the recent years, so we know about, for example, a particular kind of T-cell, a follicular helper T-cell, has specific markers, and now in the most recent classification, we've recognized those, and so now there are peripheral T-cell lymphomas with follicular T-cell helper T-cell phenotype, which we now specifically recognize. Anaplastic large cell lymphoma, another kind of T-cell lymphoma. But that name, all it says is it's really big cells that look horrible. But now we've learned over the years of some genetic abnormalities. For example, we know that ALK is rearranged, a particular gene that's rearranged that is associated with a very good prognosis for anaplastic large cell lymphoma. More recently, DUSP22 rearrangement, which is now in the new classification, also a genetic abnormality that portends a very good prognosis for these kinds of lymphomas. So that's how things evolve. It's a combination of biology and history, and that's where the names really for these lymphomas come from. So just like, I guess, to a clinician, every patient looks different, right? The face is different, the symptoms are different, the way they present is different. To a pathologist, every different lymphoma looks different. Even though you call it diffuse large B-cell lymphoma, somebody can show me a case, and I remember that case, because it looks very different than any other diffuse large B-cell lymphoma. So lymphomas look very different to us under the microscope at the patient level, just like patients look different to clinicians. We look at things like the architecture of that lymphoma. Is it diffuse? Does it have a follicular pattern? That's done at low magnification. We look at the quality and the types of cells. Are they small? Are they big? Are they irregular? Are they round? Things like that. And some lymphomas really are characteristic enough that you can get very, very close to the right diagnosis just by looking at that routine H&E alone. Hodgkin lymphoma is an example of that. But there are mimics that you want to make sure they're not, and that's why we do these other studies. There's a lot of histologic overlap and mimicry, not just between different kinds of lymphomas, but from benign things and malignant things. And so that's why we spend a lot of time training and doing all these extra studies to make sure that we're coming to the right diagnosis. So we really rely on these ancillary studies that applies our knowledge of immunology and molecular genetics to make sure that we get to the right diagnosis. So here's a slide. I like to show a few pictures. This is the kind of stuff we look at. This is an H&E, or routine hematoxin and eosin stain. And as people, pathologists are great pattern recognizers, or they're probably good bird watchers because they can see something and recognize pattern, and that's essentially what we do. And so you can see here there's these kind of big nodules across the slide, and there's like a big pink area around one of those. And then you can see little moth-eaten holes in some of those nodules, and that's very specific to a pathologist about the features. And I can say at low magnification, this looks like a case of toxoplasmic lymphadenitis or an infection, not necessarily a lymphoma. Oh, I can point it. So these are these nodules I talked about. This is that big pink area. These are monocytoid B cells. These are little granulomas, and those are features of a benign process that we know. This is a closer power of one of these follicles, and you can see here this is the center of it. There's a little mantle around it, and when we look inside, at higher magnification, these are the kind of details that we're looking at. What kind of cells are in here? There's a mixture of big cells and a mixture of small cells. That usually means something's more benign. There's little cells that have, little cells here that have little dots in them. Those are dying fragments of nuclei. That's a tingible body macrophage. That is also a feature of benignity and helps us all confirm that this is what's going on inside this lymph node. In contrast, this is actually a follicular lymphoma. So instead of having nice mantles and well-defined nodules, if you maybe defocus your eyes, you can actually see there's little follicles in here. And then inside those follicles, there's a kind of a pronounced of larger cells, a few smaller cells. They're irregular. This is a very suspicious lymph node for follicular lymphoma. And so to a pathologist, this is fairly diagnostic. But you can use additional tools like immunophenotyping to confirm that's really what it is. So many lymphomas have characteristic patterns of protein expression or antigens on their surface or inside them, and immunophenotyping allows us to detect them. We use antibodies that are developed to these specific antigens. We can label those antibodies with little markers so that we can see them, either enzymes, so we can run chemical reactions on them and make it visible, or immunofluorescence, for example. So flow cytometry uses immunofluorescence. Immunohistochemistry on tissue sections by bright field uses enzymes to make them colored so we can actually see them. So by flow, we basically grind up the tissue. We get single cells out of that lymph node biopsy. We label those cells with all these antibodies that can recognize all these molecules that we're interested in. Each of these antibodies are labeled with very specific fluorescent markers. And now we can just have those antibodies bind to the cell, and then we can hit them with laser light, look at the kinds of fluorescence that comes off it, and separate it and say, oh, yes, this marker must have been on that cell because it's fluorescing with that color. And we can label now antibodies, like up to 10 antibodies on a single cell simultaneously. We can shoot these cells through a flow cytometer at thousands per second, and then measure the fluorescence with a computer and get information about what's on those cells. And these are dot plots of a flow cytometry plot. Each individual dot is a cell, actually. And so you can look at things like the light scatter characteristics from the laser light, whether it's fluorescent with the CD10 color or the CD20 color or the Kaplan or lambda. You can see here this is then a population of cells that are expressing B cell markers like CD20 and CD10, which is a germinal center marker, and then they're all expressing lambda light chain instead of kappa. So this is a monoclonal B cell population with a phenotype that is like follicular lymphoma. And so this really tells us what's in that lymph node, and we can get that information very rapidly. Immunochemistry, this is more visual for a pathologist, so we can take a tissue section and stain it with antibodies, just like we talked about for flow cytometry. But instead of being labeled with fluorescent markers, they're labeled with enzymes, and then we can run chemical reactions on them to make it turn a color like brown, and we can then look at where those cells are and whether they're present. So this is kind of a schematic. This is another picture of the immunochemistry lab. And these are, I think this is a CD30 stain, which stains Hodgkin and Reed-Sternberg type cells. In a case of Hodgkin lymphoma, you can see how they light up with that nice brown color. This is that case of follicular lymphoma I showed you with the H&E. So now we've stained it with CD20, which marks all B cells, and you can see those nodules. Now, if you didn't know that those nodules are there, now you can see them. They're intensely CD20 positive. CD3 is a T cell marker. Those are, so you can see those nodules don't have very many T cells. They're almost all B cells. And then we can use a marker like BCL2, which is a protein that's inside cells, but that in normal germinal center B cells, you don't see that protein. There's a genetic abnormality, the BCL2 immunoglobulin heavy chain gene fusion, which causes abnormal expression of BCL2 in these germinal center cells, and now you can see it with this antibody towards BCL2. So those are the kinds of things that we do to help make the right diagnosis. Molecular studies we talked about, we can look for monoclonality. Cancers are clonal, so they are multiple copies of the same cell. And so we can look at molecular markers of clonality and do studies to show that that's what we have in that population, that they're all the same clone. We can detect those chromosomal abnormalities. We can actually, as Dr. Evans said, we can actually look at the gene. So you can manufacture probes to specific genes and make them fluorescent and then look to see if those genes are together or separate. In tissue section, we can do gene expression profiling more in a research setting, but a few companies, individuals have made some tests that can be done in the clinical lab for research purposes. And then a little bit we'll talk about high throughput sequencing. You may have heard about that, next generation sequencing, which can detect mutations that are important in diagnosis, prognosis, potentially therapy selection, and even techniques to look for minimal residual disease, which is becoming important and certainly has been important in the acute lymphoblastic leukemia setting and is making its way into lymphoma and myeloma. This is just an example of what we do for immunoglobulin heavy chain. This is the gene that makes an antibody. And what's, you can see on the top where you have a polyclonal situation represented by the different color of cells. And the immunoglobulin heavy chain gene, we can look for a piece of it. And each cell makes a different antibody, so it has a different length of part of that gene, and we can sort of size that and look at it. But in the monoclonal situation where it's all the same size, we can see this one I selected out maybe has 85 base pairs to its the part of the immunoglobulin gene that we're looking at, and we can amplify that and then size it and see that, oh, we don't have lots of sizes of this particular piece of DNA. We have only one size. That means it's monoclonal. Doesn't necessarily mean it's malignant, but it means it's monoclonal. This is just a picture so you can see. You're actually looking at some genes here now. We have a green probe and a red probe. The green probe is to the immunoglobulin gene. The red probe is to the BCL2 gene. And so in that follicular lymphoma I showed you, where the BCL2 and the immunoglobulin genes are sort of cut and pasted together. So when you stain a cell for these probes, in a normal situation, you'll see just separate red and green signals. But because the gene is fused, you get the fusion of red and green, and you're looking for yellow. So that's what the technologist, pathologist looking when you do FISH is, do I see yellow or do I see green? If I see yellow, that's abnormal. Those genes are fused. Another example would be from Burkitt lymphoma. This is an aggressive lymphoma. It's a hallmark, genetic hallmark is a MYC rearrangement, and so now you can make probes, sort of a different strategy where you have well, it's actually in this one you can do a similar strategy, having immunoglobulin green, MYC red, and again you're looking for really close red and green signals or yellow signals, and that means that the immunoglobulin and the MYC genes have been put together, which is very characteristic of Burkitt lymphoma. Gene profiling, this is that 50,000-foot view where you're basically looking for patterns. It's really essentially pattern recognition. The cases are on the columns, the genes are all the rows, and you can see some kinds of cases look very similar because they have a lot of red at the top. and a lot of green at the bottom, but they're different from other kinds. Even though the histology is the same, the genetics are different, and that's what these kinds of studies are telling us. This is, as he mentioned, is really more of a research tool, but there are people who have figured out ways to sort of reduce this to practice. You can use immunohistochemistry that I just showed you with just 3 markers and get to a pretty good decision about whether you have one type or another type of diffuse large B-cell lymphoma. This is exactly how we do it. This is a case of large cell lymphoma. You can see these really large round cells. We know they're B cells. They're marking with CD20. In this case is negative for CD10, positive for BCL6, positive for MUM1. In that algorithm I just showed you, this would be a non-germinal center B cell lymphoma, which is likely to be an activated B cell lymphoma, and so you can reduce this to practice a little bit. We and others have made some more limited gene panel tests to get at that distinction, and some of these are being used in the setting now of clinical trials to investigate whether drugs that might be better, for example, if the activated B-cell type of lymphoma will work, but of course we have to identify those lymphomas first, and these are the kind of tools that are being used. Apart from just diagnosis, there's a lot of other information that we've learned, and just to give you an example, for example, there's a molecule called ZAP70 that portends an adverse prognosis in CLL. That was discovered because of gene expression profiling studies, and we can now look for that protein by flow cytometry, for example. In the bottom corner here, you see this is a Ki67 or proliferation stain in mantle cell lymphoma from gene profiling studies. We learned that the proliferation signature is very important, and we can reduce that to a Ki67 stain, which identifies proliferating cells, and show that the high Ki67 cases do worse than the low Ki67 cases. Primary mediastinal large B-cell lymphoma and Hodgkin lymphoma were shown to be related based on gene profiling studies. Some of the particular markers could be used diagnostically. MAL is a gene that was identified as being part of the signature of Hodgkin lymphoma and mediastinal large B-cell lymphoma. We can have antibodies to that MAL protein and look to see whether an antibody like MAL could be useful to diagnose primary mediastinal large B-cell lymphoma, which could look like any other diffuse large B-cell lymphoma except it's in the mediastinum. But we could use this to sort of help refine our diagnosis, and we do that today. Something just to get towards the end, some of you may have heard about next-generation sequencing in the liquid biopsy. That's going to be something that's coming down the road. There's been research being done on that, looking at cell-free DNA. That's DNA that gets into the bloodstream because the tumor cells are dying and releasing their DNA. The living tumor cells you may be able to get DNA that they leak out into the circulation, and you can analyze that. You can isolate that from plasma and then do molecular studies and sequencing on that to understand the kinds of mutations without necessarily having to do a biopsy. So there's a lot of challenges to the liquid biopsies. You can imagine this is a very technically difficult process. There's a lot of bioinformatic management that's going to have to be done with all this information that's coming out of just a couple of drops of blood. Not only that, even if we get the right technical answers, then we need to know how do we really analyze that? What does it mean clinically? Those are all studies that need to be done. And then, of course, since I live in a clinical laboratory where there's a whole regulatory role of how do you make sure that these tests are being done correctly and the results are correct. But there's definitely been work. I mean, there's already been studies showing that you can show tumor-associated mutations in the cell-free DNA. You can see other mutations that can be detected in the cell-free DNA that's not seen in the tumor DNA. What does that mean? Maybe it's that the liquid biopsy is a better sampling because if we're looking at tissue, you only have one tiny piece of a systemic disease, and there may be other subclones in the body that you can't see from the biopsy, but maybe from the liquid biopsy would be present. You can think about predicting relapse, looking at residual disease and identifying disease progression with the liquid biopsy, and maybe even the emergence of resistance markers that would dictate changes in therapy. And just to show you sort of what people are doing now, this is a relatively recent paper that came out in one of the major journals in hematology looking at the liquid biopsy and plasma cell-free DNA. And these are basically The little dots is a particular mutation. How common it was in the blood is on the y-axis, and then across the x-axis different time points, and just showing that we can detect in cell-free DNA mutations that were in the tumor, and when patients are clinically relapsing, we can find similar mutations in the blood at the same time. And then even more provocative, in a case where you have mutations that you can detect pre-treatment at the time of diagnosis. These are some of the genes, and in this case, the mutations go away when the patient is in remission, and just before relapse in the blood they detected this mutation, which they knew to be present initially, and then a short time later, then the patient relapsed in the CNS. And then they showed that mutation also was in the CNS. And so these are the kinds of very interesting studies that are starting to be done. We're a long ways from implementing that, but I'm sure we will see this technology being relevant in the future. So in summary, modern diagnosis really requires a combination of traditional histopathology, the immunology molecular techniques that I talked about. Excisional biopsies, whenever feasible, are preferred for new diagnosis. I know we try and do things minimally invasive and do little needle biopsies, but it can make it hard to make the correct diagnosis. And as you can see, we need to do a lot of ancillary studies, and for that, we need some tissue. Pathologists like me work behind the scenes to apply all these biologic principles to patient samples to give great answers to Drs. Evans and Kelly and Sotomayor so they can select the best therapy and management plan for you. So with that, I will stop, and thank you for your attention.